Okay, does that look good? Okay. Hi, everyone. Thank you so much, everyone, for coming today. I'm going to be sharing what I've been working on for the past 5 years with you all, and I'm really excited to cover my dissertation chapters today. So the theme of my dissertation was investigating the molecular mechanisms of enhancing coral resilience to thermal stress. So coral reefs are some of the most biodiverse ecosystems on our planet. Despite only covering less than 1% of the ocean floor, they provide services to marine life, over 25% of marine life. And we also benefit from coral reefs in a lot of ways. So coral reefs are extremely valuable to us. They are economically valuable to the tune of over $1 trillion worldwide. They're also very culturally significant to a lot of communities. And they support over 1 billion people in terms of nutrition and in terms of tourism. And they also provide us with billions of dollars of support for coastline protections, where they're physically breaking down the wave energy that comes to the coastlines and protecting our communities. But not only that, coral reef ecosystems are just incredibly unique and incredibly beautiful. And they are unique in that they're constructed by animals. The framework is constructed by animals that can grow to be so large that you can actually see them from space. And just like the Great Barrier Reef pictured here. And these coral animals have diversified over millions of years to span thousands of species worldwide with diverse morphologies and life history strategies. And as I mentioned, corals are animals, but they're colonial animals, which means that they're made up of hundreds of thousands of genetic clones that are each individually called a polyp. And within that polyp is a mouth and tentacles where they're able to feed on phytoplankton or microorganisms in the water column. But despite that, they actually get over 90% of their energy source from an obligate symbiosis, their partner Symbiodinaceae, which lives within the gastrodermal tissues of the coral. And this symbiosis is extremely important to my dissertation because it can be disrupted by environmental changes, which ultimately leads to starvation and death of the coral if the environmental stress is not mitigated. So coral bleaching, this phenomenon is called coral bleaching, and it's one of the greatest threats to coral reefs worldwide. And it is driven by anthropogenically induced climate change. Sea surface temperatures are at a record all-time high. As you can see, the daily mean temperature across the years, 2023 was the highest on record, but 2024 has already surpassed all possible levels from the years prior. So we're entering this new normal of environmental change that is undoubtedly going to cause problems for coral reefs. And as Ian mentioned, we're already in the 4th global coral bleaching event. And these bleaching events are becoming more frequent, and they're spanning reefs across the globe, no matter their proximity to urbanization or the most remote reefs of the world. Nothing is safe from this ocean warming. But coral bleaching, it's not only driven by that temperature stress, the level of temperature stress, but also the duration of the stress that it endures. So as you can see in this map of sea surface temperature for Chica rocks, the gray line is the 2023 sea surface temperatures, and all of those peaks are exceeding the bleaching threshold. And we can calculate the amount of degrees that are above that bleaching threshold and calculate that as degree heating weeks, which is a metric that a lot of researchers use to compare the heat accumulation. And you can see based on this gray line that the degree heating weeks were 3 times the previous record last summer. And here in Florida, we were not immune to that heat stress event. Last summer was the worst bleaching on record, and it was so severe because from this map, you can see the buildup of heat stress accumulating as early as July. And that's a lot sooner than we normally see heat stress accumulating. And that's what drove those levels to be so high. And Florida's coral reef is not only important to us here in Florida, but it's the third largest barrier reef in the world. It's extremely important as a resource for us to conserve and protect. And Florida has been exposed to even more mass bleaching events than the global average. So it's experienced 8 mass bleaching events at this point. So there's a lot of stress that these corals have already endured and tolerated. But when we look and survey the corals during coral bleaching events, we see that corals bleach variable depending on their species, within the species or between different species, as you can see pictured here. And so that variability provides sort of a glimmer of hope and something really interesting that I wanted to study in understanding what's driving that variability for one coral to bleach over the other. And so there's 3 key parts that I wanted to cover for what would potentially drive this bleaching variability. So first is the species themselves or the population, accruing genetic adaptations over time to be more adaptable to that high temperature. Also, the algal symbiont is extremely important in determining whether they are able to bleach or more resilient to temperature stress. But lastly, the environment is also super important. So here is a picture of a map of American Samoa, and you can see that these 2 tidal pools, the highly variable pool and the moderately variable pool, are very close in proximity to each other, but one is experiencing much higher temperature fluctuations. And corals from that high variability thermal tidal pool are actually more resilient to bleaching. And so this was the environment as well as symbionts and species, they dictate these different bleaching outcomes. And so it can be very difficult to predict in these mass bleaching events. But my dissertation is really focused on the role of environment in this bleaching variability. And so the surrounding environment can influence certain traits. So we call this plasticity when a trait is able to change or shift based on its environment. And one example I'm giving here is plants of the same species grown in different temperatures have different leaf composition, leaf shape, leaf coloration, and the number of leaves. So this trait is determined as plastic because of that. And when that plasticity becomes beneficial to the organism, that is acclimatization. So it shifts to a more positive outcome for that organism. And that is not unlike when you are going to hike, say, Mount Everest, you would want to go acclimatize to that new altitude first. So you would show up a few days early and your body would adjust to that. So plasticity is a really important component of this bleaching variability. And a lot of research has gone into this. So acclimatization via preconditioning has been studied extensively in the laboratory. And we can see that corals are able to become more tolerant to heat stress where they don't even bleach at high temperatures based on the pre-exposure that they are given. And while there's a lot of literature looking at this, the field component is still lacking. So my dissertation is driven by this gap in knowledge of, can we use acclimatization as a tool to benefit corals, specifically in a restoration context? So when I say coral restoration, I'm talking about coral restoration because that is one of the main tools we have at our disposal currently to rebuild the degraded reefs, particularly here in Florida. And so we can actually take advantage of the colonial nature of the organism and fragment and grow these corals to outplant and rebuild this reef. But unfortunately, they are not immune to the same stressors that ultimately cause their demise. And so the long-term survivorship of these outplants remains uncertain. So the questions that are guiding my dissertation research are, first, can we leverage acclimatization to influence or enhance coral thermotolerance? And if so, what are the underlying mechanisms of that acclimatization? And lastly, will they persist over time? And will we see that this can be incorporated into the coral? So for my first data chapter, I investigated the role of the environment in shaping coral thermotolerance. My second data chapter, I then built upon that and looked at transplantation to that urban environment and looked at the molecular mechanisms involved. And lastly, I returned to the laboratory where I looked at the use of this acclimatization technique to more specifically pinpoint what is happening at the physiological and genetic level for two species of reef-building corals. So for my first chapter, I first characterized sites for thermal priming, and then I evaluated survivorship of corals from the reef and from the urban environment to see how they would perform in their non-native environments. So in Miami, I guess luckily or unluckily for the corals, there are abundance of stressful environments in the marine ecosystem here. And so this is due to, the proximity of human populations to the coastlines, but also that shipping traffic and that daily amount of waterway use that is occurring. But despite that, if you look even within the port where those cruise ships are parked, there are corals growing on the seawalls and the rubble and just every surprising part of that environment. So corals are present among trash, even growing on trash, but also growing to large sizes that we don't actually see as often on the reef anymore. And so a lot of research has recently gone into trying to understand why that is and why they are doing so well in the port. And so when we picked 4 different sites in the port of Miami, and we compared them to an offshore reef called Emerald Reef, researchers found that the diversity of species was actually greater at all of the port sites compared to the reef. But not only that, when they characterized the environmental parameters, they found that temperature, salinity, and pH were all super variable in comparison to that reef site. And not only were they more variable, they were reaching these extreme levels that we don't see on the reef. And when one study tried to investigate, the mechanisms that may be driving this, they found that corals from the urban environment were upregulating mechanisms related to the stress response, the immune response, and also heterotrophic activity, which is an indication of greater feeding of those corals in the port environment. And so these were hypothesized to be those mechanisms driving resilience in the port. So for my chapter, I wanted to build on that research and ask, do these corals from the urban environment maintain their resilience once you transplant them offshore? And conversely, do non-native corals transplanted to the port of Miami, will they become more stress hardened because of exposure to that more variable environment? And so for my first chapter, we picked 2 sites, one urban site and one reef site. And we deployed instrumentation at both sites to characterize continuous environmental parameters such as temperature, pH, salinity, current speed, light, and depth. And then for the corals for this experiment, we collected 3 urban native colonies of Pseudodiploria strigosa and 2 reef native colonies of the same species, and then fragmented those into biological replicates for the reciprocal transplant. And so for that transplantation design, we have 2 experimental directions. So first are the urban corals transplanted offshore to the reef. Then we have the reef corals transplanted to the urban site. And then we have 2 control groups where we're just assessing the effect of transplantation itself, but we're not actually moving them to a new environment. So for the experimental timeline, this began in December of 2022, where we collected the corals and also collected tissue samples for initial genetic analyses. And then we returned in January to monitor the health status of the corals, again in April to do the same thing. And then in July, this coincided with that mass bleaching event last summer. And so we took tissue samples at this point to get a signal of what was happening at the genetic level. And lastly, we returned in November to see how those corals recovered and also took tissue samples. So for the first result, the temperature profiles of these 2 sites over the past year, unsurprisingly, the urban site had much more variable temperatures across the year, but the summer was much hotter and the winter was much colder than the reef site. And now putting this back into degree heating weeks, I first want to set the stage for what NOAA's Coral Reef Watch sets as their sort of initial levels of thermal stress. So the 4-degree heating weeks would be a risk of bleaching, and then 8-degree heating weeks would be a likelihood. So the reef site exceeded that 8-degree heating weeks, but the urban site was almost over double that level of heat stress. And when you look at NOAA's new bleaching alert levels, which they had to recently create because the heat stress has been getting so bad, the urban site was at the level 5, while the reef site was at a level 2. And so this urban site was experiencing one of the highest temperature stresses of the area. And not only that, the seasonal and daily temperature variation that those corals were experiencing was much greater at the urban site compared to the reef site, as you can see on this radar plot, which is showing the proportion of the intensity or the, I guess, maximum values for each site. And one thing that might be driving this higher variability is the current flow and direction, the intensity of currents that the urban environment is experiencing versus the reef environment is much higher at the urban site. And in terms of other environmental parameters, salinity and pH were both significantly lower at the urban site, showing a more stressful environment. But if you can, as you can see with the height of the box plots themselves, they're also more variable based on the range of values that they can experience. And so overall, I think I've convinced you all that the urban site is much more extreme than the reef site in terms of the environmental conditions. But what about the coral outplants themselves? So these are photos of just one representative coral outplant per transplantation direction, and also the temperature that was recorded at the time the photo was taken. So as we can see, through January and April, we just maintain healthy tissue coloration. But in July, we obviously had the bleaching event, but the bleaching levels were different. So at the urban site, we had 100% bleaching across all outplants, while at the reef site, we had severe paling, but it was not to the same degree as the urban site. And when we returned in September to monitor for spawning, the corals were still 100% bleached. So that is 2 months of being in that state. And so when we returned in November, I was not expecting to find really any outplants left based on that heat stress that accumulated. But we saw 83% recovery of the outplants based on the survey we did in November. So that was really surprising and also really exciting. And when we look at the proportion of survivorship from July to November, we see that the urban corals maintained high resilience to bleaching. So they had high survivorship regardless of their location. But when we look at their survivorship across the year-long transplant, if you see that blue line is significantly lower, where they had the lowest survivorship rates across the whole year-long scale. So what's happening there? Well, I think what's happening is that high initial mortality that's occurring is due to fish predation. So we essentially outplanted these non-native corals to the reef environment, and fish, corallivorous fish, were just given a buffet essentially for that site. And so we saw really high mortality early on in the experiment, but following that bleaching event, there was high resilience. We also saw at the urban site specifically, there was a lot more pollution. So even at one point, a blanket fully smothered all of one cluster of outplants. So that took out a whole section of the experiment. So both sites were experiencing stressors, and this ultimately led to the decline of more of the urban corals, unfortunately. So returning to my questions for this chapter, do urban corals maintain their resilience offshore? So we would expect, if this were true, that they would perform, the reef transplants would perform equal to or better than their controls. Based on survivorship, we did not see that. We saw significantly lower survivorship. But based on the bleaching resilience, we did see similar outcomes. And so I think this points to potential positive result for their resilience maintaining offshore. And what about in the other direction, the corals from the reef coming to the port of Miami? Well, based on the survivorship curves, they performed similarly to their reef controls. And so I think that this points to, their survivorship will be maintained following transplantation. However, when we look at the bleaching resilience, we saw a much more drastic decline in their percent survivorship compared to their controls. And so I think ultimately this points to signs of stress occurring, that the reef corals were not as adjusted to that urban environment. But ultimately, I think the best way to assess whether the environment was stress hardening for them would be to transplant those reef corals back onto the reef and see if they perform better than reef counterparts that were never transplanted. So I think this would be a potential avenue of future research. So that was my first data chapter, where we characterized sites for thermal priming, and we also evaluated survivorship of corals at the urban and reef sites. And so for my second chapter, I built on this work by looking at the molecular mechanisms involved with transplanting corals to the port environment. And specifically, I was really interested in using Acropora cervicornis for this chapter. And that's because Acropora cervicornis is one of two key reef building species that used to be in high abundance here in Florida, but has ultimately declined drastically over the past several decades due to disease events and bleaching. And Acropora cervicornis has also been one of the main species used in coral restoration practices because of its ability to fragment and grow quickly and outplant. However, when we project the outplant survivorship in the long term, as this paper did in 2020, we see that the long-term survivorship still remains limited for this species. And unfortunately, a recent article came out that basically said that they predicted about only a quarter of the outplants of Acropora cervicornis survived the bleaching event from last summer. And so there's still a lot of work we need to do if we want to boost their thermal tolerance in these heat stress events. So one study that we published in 2022, we were able to test whether we could enhance the species' thermal tolerance. We did this in the laboratory setting using a diel variable temperature regime, as you can see in the red line, with the exposure of high temperature stress for a couple hours at a time for 3 months. And we found that the corals treated with this variability were able to endure high temperature stress double the length of time as the controls. So they were able to endure heat stress before showing signs of bleaching. And so we were able to demonstrate this in the laboratory setting, but can this be applied in the field? Because ultimately, this is not a scalable or it's a logistically costly technique to apply in the laboratory and then outplant those individuals. Can we do this in the field where it's more streamlined and easier to make part of restoration efforts? And so that was driving the questions for my second chapter, which was, can we prime nursery-raised Acropolis cervicornis in urban environments? And what are the genetic mechanisms that are involved in acclimatization? So for this study, we again returned to the Port of Miami and the offshore nursery site, which is very close to Emerald Reef. But I want to take a second to highlight the interesting site in the Port of Miami, which is the Coral City camera. And the Coral City camera is a live stream, a 24-hour live stream that you can tune in to see sort of life on the reef in this urban environment. And you can also see how healthy the corals are despite being in the Port of Miami. And it's a really great outreach and citizen science tool that I really wanted to incorporate in my dissertation because of its just innate value and also just the amazing things that it's been able to capture. So for this experimental design, we collected corals from the Rescue Reef Key Biscayne Nursery and then outplanted them to the Coral City camera site in June of 2021. And then we returned in late October to collect tissue samples from the Coral City camera site and nursery counterparts to compare the effect of location on the gene expression. And so when I'm talking about tissue samples, I think one thing that's really important that we have in our disposal for research with corals is we're able to take these snapshots in time and try to understand what is going on at the molecular level. And so to do that, we can collect small tissue biopsies and actually look at the presence of mRNA in their cells. And we can target that to understand what genes are being expressed at a given time based on the environment they're in. And so for this experiment, I collected 20 tissue samples. I ran RNA extractions and sequencing and the bioinformatics pipeline that you see on the screen, which ultimately got me the number of gene counts that I could then analyze to understand how environment influenced their gene expression. But first, just to take a snapshot of what temperatures they were experiencing at this time. In 2021, the heat stress was not as severe, but the Coral City camera site did exceed the 4-degree heating weeks. That is one of the Coral Reef Watch indicators. And the Coral City camera site is again more thermally variable than the offshore nursery. So to look at the gene expression patterns, I want to walk you through this figure because this is a way that we can analyze global gene expression patterns in two-dimensional space. So a principal component coordinates analysis has on the X and Y axis degrees of variability in which you can measure based on the proximity of a sample to another sample. And so the green samples are more similar in their gene expression patterns, and the yellow samples are more variable in their expression patterns. And so what we can see with my samples is that the nursery corals in blue were significantly more clustered to the left, while the Coral City camera samples were more clustered to the right. So we did see an effect of location. But we can also look at the genes themselves and try to understand what mechanisms are going on based on the gene expression patterns. And so for this plot, this volcano plot, we're looking at significant expression of genes based on whether they were more upregulated based on the mean level of gene expression and targeting the ones that are more significant. So for the nursery corals, they had more significant upregulated genes. And for the Coral City camera corals, there was a higher number there. And so we can actually look at the identity of these genes and try to understand what is going on at the molecular level. So in this table, you can see that in the Coral City camera corals, we have more immune response, stress response, and heat stress related genes, while the nursery corals have more cell signaling patterns. We can take this a step further and look at genes that are expressed in similar ways, or they have similar patterns, and we can correlate those to traits to try to understand how gene expression relates to the traits we observed. So this is one column of the heat map showing the correlation score, and only the significant scores have their number in the square. And so we end up with a certain number of modules that are consistent of a different number of genes. And you can see the bar plot on the right is showing the number of genes in each module. But on the x-axis, you can see we have different temperature parameters that were calculated based on the temperatures they were experiencing, as well as the site outplant or the site that they were collected from themselves. And so from this, we can see that there are two modules that are clearly significantly correlated to all of these parameters. In those two modules, we see, again, signatures of genes that are highly upregulated in the Coral City camera corals that are related to apoptosis, the heat shock or the heat stress response, as well as cellular metabolism, as in the case of cytochrome P50. But interestingly, cytochrome P450 was also highly upregulated in the corals from that highly variable pool in American Samoa. So that was an interesting finding to find overlap in terms of determining whether this was involved in a thermal acclimatization. So for this chapter, based on the environmental parameters they were experiencing at the Coral City camera site, and based on their outplant survivorship in that first year, the corals were primed to this more variable environment. But ultimately, in that 2023 mass bleaching event, we did lose all of the outplants. And so obviously the level of heat stress really matters for whether for their long-term survivorship, and the level of priming they experienced was likely not enough to cause an acclimatization signal in the long term. However, one glimmer of hope you can see in the photo on the right is that one Acropora cervicornis that is not bleached at all. And that Acropora cervicornis is actually native to the urban environment and hosts a different type of symbiont than all the other outplants. And so that is certainly an avenue for exploring acclimatization of the species to the urban environment. And for summarizing the gene expression results, we did see signals of acclimatization at the genetic level in terms of heat shock proteins, apoptosis, and cellular metabolism. And so that was the findings from chapter 2. But for my last chapter, I really wanted to hone in on the molecular mechanisms underplaying that thermal priming. And I wanted to do this using two species that are propagated in restoration efforts, one of which is Acropora cervicornis again, but another which is Pseudodiploria clivosa. And not only that, I wanted to look at the physiology differences based on that priming treatment, and look at whether that treatment had signals that showed it was maintained in the long term. So like I mentioned, we had that publication in 2022 where we did show that thermal priming of Acropora cervicornis worked. However, the treatment itself was 3 months, like I mentioned, which is a very long time if we want to scale this up and apply this to more corals. Additionally, we did not do a follow-up heat stress assay to determine whether that signal was prolonged. And lastly, we did not take samples for molecular analyses. So these are all things I wanted to incorporate in my chapter. And for the molecular mechanisms of acclimatization and thermal priming, because we do have a whole suite of research that has looked at this, the studies are not in congruence with one another because it really ultimately depends on the level of preconditioning that you apply, as well as the level of heat stress that you apply to invoke that response and assess whether their bleaching tolerance improved. And so as you can see in this figure, based on the duration of the preconditioning, you actually have two different genetic responses occurring. And so this is causing this is pointing to a gap in literature where essentially we still don't understand what is driving thermal priming at the molecular level. And like I mentioned, while Acropora cervicornis is propagated widely in restoration efforts, we have at our disposal and have incorporated more boulder mounding brain coral species in restoration, but we don't know as much about their thermal tolerance or whether they can be primed using these acclimatization treatments. And so with that, I wanted to address in my chapter what are the immediate physiological effects of thermal priming, what are the immediate molecular effects of this treatment, and then what are the long-term physiological effects. So again, we return to the Port of Miami and to the coral nursery. And the Clivosa colonies were collected from Star Island in the Port of Miami, and then 3 genotypes of Acropora cervicornis were collected from the coral nursery. And they were all fragmented and subjected to that same temperature regime, but this time it was only applied for 28 days. And so I'm going to walk you through the experimental timeline for this in this figure. So the treatment was part 1, and during that we recorded different physiological metrics that you can see in that table below. But following that, the first heat stress assay we used was a short-term heat stress assay, and we mimicked temperature profiles from a more recent metric or a more recent tool called CBAS, which is the Coral Bleaching Automated Stress System, where you can essentially assay the coral thermotolerance in less than 24 hours. So we were able to do this rapidly and measure the coral's algal photosynthetic efficiency as a proxy for their thermal tolerance. And then the corals were given 60 days to recover from that heat stress assay. And following that, they were subjected to a long-term heat stress assay where one temperature was maintained for 3 weeks. And during this time, we recorded survivorship and bleaching and disease incidence. And for the genetic sampling, we took tissue samples from both species at the beginning of the treatment and then at the end of the 28 days. And then I ran that same tissue processing and bioinformatics pipeline to get the gene counts for this chapter. So I'll first start with the physiology from the treatment portion. So based on the calcification rates and the tissue coloration we observed through photographs, we did not see a significant effect of growth or tissue coloration based on the treatment itself, which was good because we didn't want to see that the treatment was negatively impacting the corals physiologically. However, the coral symbionts were affected by the treatment, but it was different based on the species. So for cervicornis, the variable temperature treatment actually benefited the algal symbionts, where they didn't have as drastic of a decline in their efficiency. Clivosa was the reverse, where the treatment itself actually caused a problem for their symbiotic efficiency, or photosynthetic efficiency. And now looking at the gene expression patterns from this treatment period, we see that only for Acropora cervicornis were the samples separated distinctly based on treatment. For Pseudodiploria clivosa, there was no clustering based on a treatment or time point. And so for the rest of this chapter, I'm just going to be focusing on the gene expression results from Acropora cervicornis. So looking at a volcano plot again, we see over 1,500 genes were differentially expressed between the treated and untreated groups. And when we look at some of the gene annotations from these, from that volcano plot, we see some really interesting genes that I want to highlight. So the first one is a significant upregulation of histone 2H2A. And so this is actually a marker for epigenetic modifications where we can, at the gene level, histone markers basically influence the accessibility of the DNA. And so that ultimately affects transcriptional regulation depending on what markers are present. But not only that, in 2019, this review paper showed that a bunch of different histone modifications are associated with thermal acclimatization. And so this marker was really interesting to find in our data set because it's supported by a lot of other literature connecting it to thermal priming. The second gene of interest was nitric oxide synthase activity. And so this was actually significantly downregulated in the treated group compared to the untreated. And nitric oxide is a signal of, it's involved in the oxidative hypothesis of coral bleaching because it ultimately triggers the production of more reactive oxygen species at the cellular level, which is a cellular stress response. And if those accumulate, it can ultimately lead to the breakdown of the coral symbiosis with the algae. And so the fact that this synthase was suppressed in the treated groups suggests that it was able to downregulate production of this molecule and ultimately prevent the accumulation of reactive oxygen species. So the last part of this study that I'll talk about today is the results of the long-term heat stress assay. So based on the treatment that occurred, that 28-day treatment, I then plotted the survivorship of both coral species based on their initial treatment. And you can see here that Pseudodiploria clavosa had significantly higher survivorship in thermal stress, on the order of 15 or more days longer. But not only that, the treated corals had higher survivorship than the untreated group. Acropora cervicornis also showed this trend, which was really exciting to see that the treated corals were still enduring heat stress significantly longer. And there was that additional benefit of this study to comparatively show that the species had significantly different bleaching or heat stress or heat tolerant thresholds. So what can we say about the physiological effects of thermal priming? Well, growth and tissue coloration were not significantly affected, but photosynthetic efficiency was. In terms of the gene expression, the gene expression patterns I talked about do indicate a response of thermal priming at the molecular level. And probably the most exciting part to me was to see that the treatment that occurred at the beginning of the experiment, 4 months later, we were still able to detect a signal of enhanced thermotolerance. And so for my third chapter, I was able to address my aims for both species and identify more molecular mechanisms involved in acclimatization. So bringing all of my chapters together, how do they tie back to these three questions? So first, can we leverage acclimatization to enhance coral thermal tolerance? Well, based on my first chapter, I demonstrated that the highly variable urbanized environment acted as an environment to create more thermally resilient corals that ultimately maintained that resilience offshore. In chapter 2, I showed that transplantation to the urban environment did lead to signals of acclimatization at the molecular level. And in chapter 3, I showed that the variable temperature treatment also led to increased survivorship of those treated corals. But what about the underlying mechanisms involved? Well, a lot of these mechanisms are actually also involved in just the general coral heat stress response at the cellular level. And so I think what's important is that sublethal component, or that dosage component of temperature when you're trying to do thermal priming, is to basically trigger this response at the cellular level, but not to a point where it can go to the point of no return, and the coral ultimately becomes too stressed and succumbs to bleaching. And the last part of this, will they persist over time? In chapter 1, I showed that even after 6 months following transplantation, the coral survivorship following bleaching was still maintained. And in chapter 3, that the long-term heat stress assay did show the effect of thermal priming even 4 months later. And so to build upon this work and to take have some take-home messages here, thermal stress hardening does work for these species that I studied and the genotypes that I used, but the scalability of this is obviously something that needs to be explored further if we are to implement this as a tool for restoration. And so what I can say to restoration practitioners right now is, I think looking for more variable environments to potentially use in nursery propagation or short-term holding sites could be a really interesting avenue to potentially thermally prime some of these corals for restoration. But lastly, it's really important that we continue to evaluate the long-term feasibility of acclimatization, and whether these signatures of thermal priming will last through subsequent summers and more thermal stress events. Okay, so now I'd like to take a couple minutes to acknowledge a bunch of people and my funding sources. So thank you all for listening, and this will just be a couple more minutes. So first, my funding sources. I want to thank NOAA Omics Initiative, the NOAA Coral Reef Conservation Program, AOML, the University of Miami Rosenstiel School, and CMIS, that is Cooperative Institute between NOAA and UM, and the Cnidarian Immunity Laboratory for all of their support in these experiments. And I also want to thank the external fellowships I received while I was a student here, the David Rowland Fellowship and the Student Travel Fund, for helping me talk about this work at conferences.